Video on RNA and protein synthesis. RNA and protein synthesis. So in the processes of protein synthesis, you have the DNA, and obviously that is what we are looking at here. We know this is a DNA strand because it has thymine in it, and it also has the 2 strands. And if we were looking at it in a different dimension, it would have that double helix shape, that little twisted staircase type of, twisted ladder type of look. But this is a DNA, the structure is DNA. It's inside of the nucleus of a cell, and it can't leave. And because it can't leave, the cells need to use the DNA as a template, okay? And of course, in order to get the code from DNA, we have to have the help of enzymes. Okay, the particular enzyme that is going to help us get this code copied is going to be RNA polymerase. Okay, so that RNA polymerase is going to unzip the DNA strand inside of the nucleus so that we can create a messenger RNA strand. Now, RNA is very similar to DNA. One of the differences is that instead of thymine, RNA is going to have uracil. So uracil, it pairs with adenine instead of thymine when we're looking at RNA. So we're going to go ahead and we're going to use our RNA polymerase to pair up our base, to use that base pairing rule to pair up our nitrogen bases. Okay, so again, guanine pairs with cytosine, and adenine pairs with uracil in an RNA strand. Okay, and I like gizmos because they color code it for us, so it's nice to have that color code, that nice visual. All right, so I'm building my nucleotides and I'm pairing up my nitrogen bases that hold the code to the protein. Okay, and so I have created here a new strand of, or I've created a strand of messenger RNA. Okay, when we continue, okay, that messenger RNA is small enough to leave the nucleus of a cell, and it goes to our ribosomes. Of course, our ribosomes are the site of protein synthesis. They're freely floating, they're attached to the endoplasmic reticulum, and they're there to work with the transfer RNA. A transfer RNA, which kind of look like chess pieces here. That transfer RNA is going to work with the messenger RNA in order to assemble amino acids. Okay, so in order to do that, it has you have to match the complementary transfer RNA anticodon, which is here, to the first codon in the messenger RNA strand. So what is a codon? A codon is 3 nitrogen bases that are going to code for an amino acid. So we need to find the transfer RNA piece that has the anticodon, which means then an A again pairs with U, a U pairs with A. So UA, so far it's looking like this is going to be our anticodon. And here this is the amino acid on that tip of the transfer RNA. C pairs with G. Okay, so we have our transfer our anticodon there. And then obviously this one's the last one here that is going to allow us to start building a chain of amino acids. Okay, so we're building this chain of amino acids. All right, and then we're putting our release factor. And after the release factor is added, we have, and this is a very small chain. Usually a chain of amino acids, it becomes a polypeptide chain, and polypeptides are those are put together to form long chains that are folded in unique ways to create protein. Okay, and so there we have what represents a new protein. So the new protein is enclosed in a membrane, and then it's transported to its target within the cell. And that is how protein synthesis works.